The next lecture, Russ Poldrack from Stanford University, hardly needs an introduction in this context. I will just mention that this is the 10th time that we're doing this and the 10th time that Russ has come to give a lecture, and we're always very excited to hear what he has to say about reproducibility, about technical things, and the broader context of this course, and I'm particularly excited this time.
Thanks. Nice to see you all. Actually, let me turn on the camera so the folks online can see. So yeah, it's always fun to be here, and nice to kind of kick things off. I'm going to be giving another talk tomorrow morning about AI-assisted coding. It'll be jumping a little more into the details. Well, the goal of this talk is really kind of a 30,000-foot level overview of like why should you come spend 2 weeks doing the stuff that you're going to be doing here, and try to motivate that that one of the reasons is that that there are problems in science that we think that the kind of things you learn here can help solve. So if you go on the NIH website, you can search for all the grants that have been funded. And if you search for fMRI, you basically find about 1,800 grants funded in fiscal year 2024. Almost $1 billion worth of total cost went into that. That's a lot of money that the federal government's putting into paying for, presumably not every one of them did fMRI research, but a lot of them did, right? So even if it's just half, that's $500 million. And if you look at the text that's associated with those things, you see stuff about brain change, treatment, clinical. Clearly, people are motivating the spending of all this money by saying that, that these tools should help us better understand how to fix problems with mental health or other brain diseases. And so we can ask whether that's actually happened. And so this is actually from a few years ago now, but I don't think that that the situation is markedly different now. This was it was kind of like a re-meta analysis of a number of studies. So, what you can do is you can go into studies and pull out the activation coordinates that have been reported, and then, try to ask how consistent are the results across different imaging studies. So several previous meta-analyses of studies of depression, basically compare people diagnosed with depression to people without a diagnosis, had claimed to find consistent differences in brain activity in task fMRI studies, for those groups. Mueller et al., who are one of the world's best groups in fMRI imaging meta-analysis, redid this and fixed some problems with the previous meta-analyses, and reported, across 99 studies that went into their meta-analysis, there was no convergence basically across any of those studies. So basically you have 99 studies all claiming to have found differences between people with and without a diagnosis of depression. Put them all together and you get nothing. That seems bad, right? We've seen other examples of things gone wrong. Now increasingly people want to apply fancier machine learning tools, and so there was a very splashy study. This was from the NIH kind of like news release thing. Machine learning identifies suicidal youth. And this shows the brain activation pattern for looking at the word death and comparing people who had tried to commit suicide versus not. And the group got an almost $4 million grant to pursue this work. This is a group at Carnegie Mellon. This paper was retracted after researchers dug into what the group had done and saw that they had basically done something that's a very common problem in machine learning studies, where the data, some aspects of the test data got leaked into the training data due to particular sort of types of feature selection that they used. So clearly there's major problems in the field. And so the question is, how do we fix this? And some of us have had this intuition for a while, kind of inspired by this quote from this famous US Supreme Court justice. Sunlight is said to be the best disinfectant. If we want to fix things, one way to go about doing that is to try to kind of make things as open and transparent as possible. So, my group, along with a bunch of others, have been involved for the last decade in basically trying to build an ecosystem for doing open science, doing reproducible science. And I'll talk about some of the things we've done, not in a lot of detail. I'm mostly going to spend my time today kind of motivating, what all the problems are that we need to be able to solve. But the goal in part is like building tools, not just to kind of gripe about problems and wring our hands, but to build tools to help people do better science. So I like to think about this as sort of like a design problem. Does anybody know what this thing is? It's a commercial paper cutter. So, you have a big thing of paper you want to cut in half, down to a different size. You see it has two green buttons on it. Why do you think a commercial paper cutter has two green buttons on it? Yeah.
And get them chopped off.
Exactly. Yes. So you will not do the thing that humans are likely to do and like try to hold the paper in with one hand while you cut with the other. You have to hold both hands down here so you don't cut your fingers off. That's a good design for a machine, right? It anticipates the stupid things that humans will do and prevents them from doing it. And that's really, I mean, if you read, books like Don Norman's Great Design of Everyday Things, design is all about basically building affordances to get people to do the things you want them to do and prevent them from doing the things you don't want them to do. And humans are, in science, the thing that we have to worry a lot about is the fact that people are really good at fooling themselves. Particularly once we have an idea, we start to look for evidence in favor of that idea, and we become blind to evidence that just confirms that idea. So one of the ways that people in behavioral economics talk about this is in terms of what we call choice architecture. The idea is that whenever you go into any situation where you're making choices, some of those choices are more likely to be, sort of encouraged than others, right? If you walk into the grocery store and the fruit is like way back in the corner and the candy bars are right up in front, you're more likely to buy the candy bars, right? And in the context of what are called nudges, behavioral economists have talked about the idea that we can use various types of features to try to get people to do the right thing, like changing incentives. Obviously, incentives are powerful, but also using the power of defaults. It turns out that whenever there's a default option for something, people are very likely to use it. So you want to give them the right defaults, providing feedback, expecting and preventing errors, like we saw with the green buttons. And so the hope is that we can use some of these ideas to try to help people do better science. How many of you have heard of or read this paper? I encourage you to all go read it. It's 20 years old now, published by John Ioannidis. And basically, it argues what the title says. Most published research findings are false. And there's been controversy about whether it really shows what he claims it shows, but I think it's a reasonably compelling argument. And one of the things he does in the paper is he walks through like, what are the features of a field of science that would make it more or less likely for the research findings to be true? And he says, the smaller the studies conducted in a scientific field, the less likely the findings are to be true. And it turns out that in neuroscience, I mean science in general, but particularly in neuroscience and psychology, research overall is underpowered. So for those of you who don't remember what statistical power is how likely are you to find a result if the result is actually true? So like, if there really is a difference in brain function between people with and without a diagnosis of depression, I go do a study of it. What's the likelihood that I will find that difference? And in general, we want that number to be at least, say, 80%. These are a couple of surveys. The one on the left is specifically from human structural imaging studies and like rat neuroscience studies, basically showing that the majority of studies are badly powered, with a lot of the studies having less than 10% power. That means you could run the experiment 10 times, and even if there is a true effect there, you're only going to expect to find it once. And then Marcus Munifo and his group have also expanded this out to kind of biomedical science more broadly, and you see that it's a general problem. Lots of really underpowered studies. If you were to tell an IRB that your study has 10% power, they would come back and tell you that you can't run it because it's what they call a futile study. You're putting your subjects at risk, even if you're just like, making them drive into the building. And it doesn't make sense to put your subjects at risk if if your study is basically futile. Now, there's another way to think about this. One argument that you might get is, well, we're controlling our false positive rate, right? We're alpha is p less than 0.05. We do our test. We're only going to find a false result, 1 out of 20 times. And that's true, but that might not be the number that you really care about. Another way to view this, and this is basically what John Ioannidis laid out, is that we might want to, instead of asking like, what's the proportion of false positives over all tests, we might ask, what's the proportion of what we call false discoveries, of what's the proportion of times when you say that there is a significant result when it's actually false. And we call that the positive predictive value. And what Ioannidis showed, and Button et al. Kind of do a nice little visualization of this, is that when you have low power, basically the majority of significant findings are going to be false. So here, I like to use thought experiments. So let's say that you're a physicist and you're trying to detect some signal out in the universe. Unbeknownst to you, your detector's broken and is giving you back basically noise. So let's say you go do 100 experiments and you're going to get 5 significant results out of those 100 experiments. How many of those are true positive results? 0, right? None of them, because your machine is just giving you noise. So your false positive rate is 5 out of 100, right? Because you've only said yes 5 times. But the positive predictive value, the number of those things that are true, is 0, right? And let's say you fix your machine and there really is a signal out there. The better your machine gets, the more likely you are to find that. So your positive predictive value will go up. The other thing that we know is that when you're underpowered, the only effects that you can find are really big effects. And so if you look at the published literature and say, what's the effect size that people are talking about in the literature? Those systematically overestimate the true effect size. We call this the winner's curse. Now, the final thing that we know is that when you have small samples, the variability of your statistical estimates gets big. And you particularly see this with correlations. This is from a while ago from Felix Schoenbrot. They looked at this thing they call the corridor of stability. The idea is, how big does your sample need to be before the correlations basically become kind of, stable enough that you can start to make something of them. And so this is showing, this is just simulation, right? Showing that like when your samples are small, the true correlation here is like 0.26, but you can find estimates anywhere between minus 0.25 and like plus 0.6 something, right? So just in different samples, right? So it's just sampling variability. You pull out different small samples, you're going to get very different results. Sorry, these are kind of small. Oh, I should say, did you put the link into the Slack? The slides are all online, so if you want to kind of follow along, then you can kind of look a little bit more closely. Some of you may be familiar with this paper by Marek and colleagues, which got a lot of play, made a lot of people very upset, continues to make people upset. And basically, it's funny, I was a reviewer on this paper, and the first thing I wrote in my review was, it doesn't really tell us anything that we didn't know from Felix Schoenbrot's simulations 10 years ago, but it was I thought it was a useful, they did more than that, and I thought it was a useful paper. But basically, they took data from a number of big studies, particularly from ABCD, and they looked at what happens when you look at correlations between brain and behavior across lots of different voxels. They did this with cortical thickness and with resting connectivity. And basically, they showed that when you use small samples, this is showing down at the bottom that when you have a sample size of 25, when the true correlation is in this case, the first one is like minus 0.08. You can get one sample that says, oh, it's positive 0.5. You can get another sample that says, oh, it's minus 0.64, right? That's simply the variability due to small samples, especially with correlations. And it's not until you get into the thousands, really, that you hit that corridor of stability where if you take multiple samples, you're going to see similar answers. And there's, other people have done similar analyses where it really does seem to be the case that these associations are just really not robust with small sample sizes. Another place that this worked out in a bad way was in the case of what people now call candidate gene studies. So back around in the early 2000s, it became possible to measure someone's genotype for particular variants, particular polymorphisms in the genome, right? So, for example, one that was popular was a polymorphism in this BDNF gene, brain-derived neurotrophic factor, which is known to be expressed in places like hippocampus. And it turns out that people have, different versions of that can cause them to have different amounts of BDNF. And so studies started taking small samples of people, say, 50 people with the high function version and 50 people with the low function version, and compare them on, say, hippocampal volume. And there were a bunch of papers that claimed to have found associations between BDNF polymorphism and hippocampal volume. This is a plot from a meta-analysis looking at what happened over time, where it's interesting. The size of the dot, I should have the numbers. The size of the dot is the sample size. So you can see basically early on in time, there were, almost basically pretty much everybody was reporting positive results from small samples. Over time, samples get bigger, reported effect sizes get smaller, and you see in this case, it tends down towards 0. We call this the decline effect. In general, it's true that later studies find smaller effect sizes than early studies. We now know that from very large samples, there is no effect. There's no relationship between BDNF and hippocampal size. There's also no relationship between a bunch of other candidate genes that people had studied and reported in the literature and confirmed in meta-analyses that, that those effects existed. There are other genes where variation does relate to hippocampal volume, genes that people hadn't thought about. So how well powered are fMRI studies? We first published this in a paper back in 2017, and then have updated this meta-analysis, just basically finding papers in PubMed and that mention fMRI and looking at their sample sizes. And the median study from last year had 36 subjects per group. And this is, you see it's been going up over time. This is actually a log scale, so it's actually going up faster than this makes it look. What we can then do is basically say, what is the effect size that each study had sufficient power to find, right? So your power is directly related to the effect size you're looking for. If I tell you, I'm going to go do an experiment to compare the height of 10-year-olds and 18-year-olds, that's a huge effect size. I don't need a very big sample to do that. I need a sample of like 4, I think, in each group to be able to 80% of the time find a significant difference between 10-year-olds and 8-year-olds. Sorry, and 18-year-olds. If I want to find the difference between 17-year-olds and 18-year-olds, I need a much bigger sample because that's a very small difference. And so when we talk about power, we need to make sure that the effect size that we're powered to find is a plausible effect size. And the question is whether that's true for fMRI. So what Joka Durnez, who was a statistical power person in my group, did this analysis that suggested that the median study in 2024 could find a single activation with a Cohen's d, which is like how many standard deviations are the two things apart, of about 0.67. That's a fairly big effect. The question is that plausible? Should we expect to find that for fMRI effects of interest? Now, we can't look at the literature and ask what effect sizes have people published, because those suffer from that winner's curse. Right? We found them to be significant via some kind of thresholding, and we know that probably is in part due to statistical noise. So what we have to do is find unbiased effect sizes. So what we did was we took the Human Connectome Project data, which has a bunch of different tasks. We created group maps, and then we found a region of interest by combining what the HCP group had written in a paper about what the regions of interest were for each of the tasks that was written down before the data were collected, and then went to neuroscience to get basically a region of interest, a part, within whatever this anatomical area was that they mentioned. What are the voxels that we're pretty sure should have an effect? of say, motor activation. And then we can use that region of interest, go back to the Human Connectome Project data, pull out the signal for each person and ask, what is the effect size? Now, there's two different ways to look at the effect size. One is like, what's the average effect within a person? Let's say I have you start doing a motor task and I look at the effect in your motor cortex and ask, how many percent does that signal go up? In general, in fMRI, you never see signal change above, on average, above about 5%. And that's what you would expect in like primary motor, primary visual sorts of cortices. And on the left here, if you look, we do see, for example, in the supplementary motor cortex, we see about a 4% signal change. So this is saying that these are pretty whopping signals, but that's not the effect size that we care about. When we're doing statistics, we have to ask, how big is the signal relative to the variability across people? That's what Cohen's d measures. And that's what we computed here. And you can see that like this is small, medium, and large effect sizes. So that whopping activity in the supplementary motor area is associated with a Cohen's d that is roughly what these studies have power to find, about 0.6 something, 0.7. So what that's saying is that the median fMRI study published last year only had power to find the most whopping fMRI signal you would ever expect to find. Realistic fMRI signals are usually probably 10 times smaller than this, like point something percent, not several percent. So if we go back and look at that Mueller et al. Study, and sorry, as a colorblind person, I should know better than using color to mark things out on a figure. What I've done, if you can see the color here, the ones in red are the ones that I claim have a grossly insufficient sample size. And that claim is based on this rule from Simmons and colleagues, basically saying, there's never a reason to collect less than 20 subjects in any study ever in per group. And there's, they lay out the reasons for that. And that's not saying a sample size of 20 is sufficient, far from it, but it's saying that a sample size of 20 is, unless you're comparing the heights of 10-year-olds and 18-year-olds, a sample size that small is almost never going to be sufficient to give you the kind of statistical power that you need to find robust results. And so why do we see studies with 18 patients and 16 controls? Well, one reason is that people do things like flexible termination, where they're running statistics as they go and they see a significant result and they stop. And Simmons et al. Showed that that can drive false positive rates through the roof. Now, you might hope that machine learning approaches would save us, but they don't. Gael Varquois has this great paper on cross-validation failure that goes through the variability in predictive accuracy that's estimated from using machine learning tools, and shows that accuracy also with small samples can be highly variable across samples. And so if you have small samples and you have some degree of publication bias, when people are only publishing the positive results that they find, you can get results like this. So he looked across, this is looking at several different meta-analyses that had brought together data on predictive accuracy for a bunch of different conditions. And basically you see for all of them, predictive accuracy in the literature goes down as a function of sample size. Why would that happen? Right? That would only, the best explanation for why that happens is that there's some degree of publication bias. So people are only publishing the results that work. Right? Otherwise, you should see a broad range that gets narrower over time. Instead, you just see it all going down. And anytime you see somebody claiming 100% accuracy at any sort of prediction thing having to do with, especially having to do with psychiatry, you should be very skeptical of that. In fact, there was just something on Blue Sky in this last week. A paper had come out in one of the JAMA journals for this pain biomarker claiming to have found 100% accuracy. And another group dug into it, and it seems like they were doing all the things that we know can kind of cause inflated accuracy. Okay, so I've been going around giving versions of this talk for a while now, or at least talking about some of these issues. And when I sit down with trainees, we will sit down at lunch after this. what I often hear is, well, this is great, Russ, but I can't afford to go collect 4,000 subjects for my dissertation. What am I supposed to do? And it's a problem, right? It's a hard problem. I think that, you've already heard about the massive amounts of data that are out in the world. Increasingly, a lot of you can't always ask your question that you want to ask using existing data out in the world, but sometimes you can, and if you can, you should do that. I love this quote from Nancy Canwisher: if you can't answer the question you love the question you can. Right. So it may be that you don't get to do exactly what you want in your dissertation, but you spend time building skills on a related question, and then you can hope to go on and ask the question you really want to ask down the road. And then there are a number of pivots. So I wrote this paper called The Cost of Reproducibility that you can look at, and it lays out a lot of these issues. But think about, collaborating. So that the field of genetics addressed this question, this problem, by basically having lots of people come together and contribute data. So now if you look at papers in the field and doing sort of genetic association studies across the whole genome, they often have authors because so many people contributed data. Another thing you can do is using shared data. Another is focusing more on theory or computational modeling or methodological sorts of questions, because those are going to be really important it's really important stuff to know down the road as well. And I mentioned the sort of golden age of data sharing. There's so much out there now that many of the questions that you might want to ask, you can at least start to ask using existing shared data. Not always, but sometimes. Any questions so far?
Okay. There's one on Slack, which I'll convey. Wouldn't multivariable analysis and network analysis be a solution for dissertations with lower subject size? They are often more overpowered than cluster-based or voxel-based.
Maybe, yeah. So it depends on the analysis. So the question, just for those who didn't hear it, is wouldn't things like multivariate analysis, network analyses, things like that be better because they're going to be better powered? That is sometimes the case. In fact, there's a paper that argues against the Merrick paper trying to make that claim. I think that paper is wrong because it's kind of like it's sort of circular. But there are going to be some cases where it is true that a network analysis or some sort of multivariate analysis can have more power than kind of a whole brain analysis. So if the power analysis tells you that you have enough power and you're powering for a realistic effect size, not 0.5, which is probably not a realistic effect size for most of what we do, then I think that could be a solution. Yeah. Okay, so let's talk about another issue that Ioannidis raised. The greater the flexibility in designs, definitions, outcomes, and analytical modes, the less likely the research findings are to be true. In that 2017 paper, we wanted to know just like how many plausible analysis pipelines could one come up with. This is just with FSL, just one of the packages that's out there, just for kind of, basic fMRI analysis, just asking at each step, like, what are the reasonable choices that somebody might make? We came up with almost 70,000 different workflows. That's a lot. And Josh Karp did a paper a while ago that looked at how variable are the workflows that people in the literature use. And it turns out that they're very variable. He looked at 241 papers and saw that there were nearly as many unique analysis pipelines as there were studies in the sample. Almost no two papers used the same analysis workflow. And you can guess that, like, here and here, these are the defaults in software packages, and that's why those are used heavily. He also showed that it actually makes a difference. He took one data set, a data set that we had shared, and ran it through, I think, almost 7,000 pipelines, and looked at the range of statistics across those pipelines and saw that there were regions where you could get like, an 8 standard deviation range in the results. And that seems problematic. And we think that machine learning approaches can actually make this worse, especially if there's any kind of leakage of data from test into training. And this is a great paper, mostly just for its title. I tried a bunch of things, basically just showing that if you try a bunch of things, you can basically find just by variability some pipelines that are going to work better. It's also not, I don't want you to, I'm talking about fMRI because fMRI is what I do, but it's not just fMRI. Luck and Gasplin have a nice paper that basically shows how to get statistically significant effects in any ERP experiment and why you shouldn't. Okay, so this is something that people have spent a lot of time thinking about in the last 15 years. And one of the solutions that people have come up with, that the field has come up with, is this idea of pre-registration, basically writing down what it is that you're going to do before you do it and putting it in a place where it's sort of immutable, where people can see what you're planning to do, and so they'll know if you did something different. This doesn't prevent you from doing exploratory analyses. It just makes it very clear to you what was planned and what was exploratory. I don't view pre-registration as like as like handcuffs, and I don't view it as a way to like prevent fraud or anything like that. What I view it as is a way to help people do the right thing and not fool themselves into thinking something is confirmatory when it's really exploratory. It's also the case that, every paper that we have pre-registered, we've ended up doing things differently than we pre-registered. We deviated. And as long as you're explicit about that in the paper, sometimes reviewers will get really upset about it. In general, it's as long as it's reasonable, it's not a problem. This is a really cool analysis that looks at what happened when pre-registration was implemented in clinical trials in the US. So in 2000, the National Heart, Lung, and Blood Institute, which funds a lot of clinical trials on like cardiovascular stuff, required that clinical trials be registered in this site called clinicaltrials.gov. And they looked at, trials before and trials after. Anything down below 1 here, especially anything that has a plus, is basically a trial where the claim was that the treatment is beneficial, right? And it would have a slash if it found the treatment was harmful, and it has just a fill dot if it had a null effect. What you can see is that prior to the requirement of pre-registration, most of the majority of studies found positive effects. After the introduction of pre-registration, the large majority of studies found null effects. It's not like drugs stopped working in the year 2000, right? What happened is people stopped being able to tweak their outcomes or tweak their analysis or whatever to be able to find a significant result. It makes you really worry about if you're taking a drug that was approved based on a clinical trial from before 2000. Now, pre-registering neuroimaging studies is hard, right? There's just, unlike if you're going to go do a clinical trial on a drug, you'll say, well, this is our sample, and these are there's like one hypothesis we want to test, or maybe 2, does the drug improve all-cause mortality, and does it improve a specific type of mortality or something like that? But for neuroimaging, there's just, the analyses are so complex that it's hard. Nonetheless, I think you should, that's not a reason not to do it. And what you should do is just specify as much as you can. Particularly, how are you determining your sample size? What are your inclusion/exclusion criteria? What are the primary hypotheses? If you're using anatomical regions of interest, be specific about how those will be identified. And then what's your analysis plan? If you can simulate data, that's always great. Sometimes it's hard. There's not great fMRI simulators. There is at this site a template for pre-registering fMRI study. Jeanette Mumford and I had a hand in helping them kind of improve this. So I think it's a pretty good template. But I would suggest that you do that, even if you're analyzing data from an existing project, like data from Human Connectome Project or ABCD. It's still worth writing everything down about what you're going to do before you touch the data. And in particular, thinking about, one of the things that we've moved to doing is holding out a small part of the data as our kind of initial discovery sample, where we basically take those data and torture them. We build our analyses on them, make sure the analyses work. And then once we know the analyses are working for our data, we take those and apply them to the rest, to the main part of the sample as our kind of validation sample. That's one way to address, how to deal with pre-registering something when you don't know what to do, right? So your pre-registration in this case would be a two-stage thing, where the first thing says, we're going to collect, say, 100 subjects, and we're going to use 10 as our validation, as our discovery sample, and 90 as our validation sample. And then we'll come back and pre-register the methods that we develop based on the discovery sample. There's various ways to try to do that. I'm happy to talk through that, some of that later. Okay, now the pre-registration helps prevent p-hacking, right, by telling you basically this is the analysis you're going to do, but it doesn't eliminate analytic variability, right? We could all go pre-register a study and all use different analysis workflows. So we wanted to know, Josh Karp showed that they seem to matter, but that was kind of a canned experiment. We wanted to know, like, if you you ask people out in the wild to analyze a data set, how different are their results going to be? And so we were part of a big study led by Tom Schoenberg and Rotem Botnick-Nazaire called the Neuroimaging Analysis, Replication, and Prediction Study, or NARPS, published a number of years ago, where we basically collected, Tom and Rotem in Tel Aviv collected a data set on, it was a gambling task that we had used before, 108 subjects split into two slightly different versions of the task. And then we went out to the field and basically tried to get people to analyze the data. We recruited teams of up to 3 people, and they had to agree to basically not tell anybody about their results until we lifted the embargo. And in exchange, we offered them co-authorship on the paper. That's why the paper has almost 200 authors. If you're ever thinking about writing a paper with 200 authors, talk to me. It's not easy. And we gave them the data. We gave them about 3 months to come back and give us answers. The main thing they were giving us back is answers to 9 questions that were of the form, yes or no, is there activation for some particular aspect of the task in some particular part of the brain? Is there activation in the ventromedial prefrontal cortex as the amount you could win on the gambling task goes up? Things like that. And we told them basically analyze them as if these were data from your own lab. The only restriction was that you had to use a whole brain analysis workflow. We got results back from 70 teams, and they gave us back a bunch of stuff beyond those yes/no answers, like the intermediate statistical maps, things like that. We also had them give us a detailed description of their workflow. So we were involved in a project a number of years ago that came out of the Organization for Human Brain Mapping called the COBIDOS project, which is basically kind of a best practices document. I would definitely, if you're thinking about writing a neuroimaging paper, I would definitely go look at the checklist that comes with this paper, because it lays out, here are all the things that you should describe in your paper. And so our teams gave us back these descriptions, 10 to 20 pages of their methods. None of the 70 teams used an identical workflow. But even with that 10 to 15 page thing, it was hard to tell exactly what they had done. We often, for reasons that I'll talk about in a moment, we had to go back and ask some of them for the actual files from the analyses to see how they had actually analyzed the data. So these are the results. This is like the main result. The y-axis here is what proportion of teams said yes or no. Sorry, what proportion of teams said yes. So if they all agreed, these numbers would all be 0 or 1, right? Either everybody would say yes or everybody would say no. What we see is that for 3 of them, these are ordered by that proportion. So for 3 of the hypotheses, pretty much everybody says no, about 5% say yes. For 1 of them, pretty much everybody says yes, about 85%. For 5 of them, you have 20 to 40% of teams saying yes. Same data, 20 to 40% of teams saying yes, the rest saying no. And if we look voxel-wise, if you basically get their maps of what voxels were active, the best voxel, this is for all the hypotheses, this is just showing one of the hypotheses, the best voxel was 76%, right? So only 76% of teams said, yes, this voxel is active. The rest of them, you have fewer than 76 saying it's active. We also got the full whole brain statistical map and basically did a big correlation map of those. So each row and column here is one of the teams. They've been anonymized with these codes. And the color scale is just the correlation between the maps from those teams. So what do you see here? Well, the big red blob in the top left basically says most of the teams had maps that were pretty much correlated with one another. The stuff up at the top here is the red means for this hypothesis, they said no, green means they said yes. So what this shows is that you can have two teams like these two that are very highly correlated, still in terms of their underlying statistical maps, one says yes, the other says no. So clearly something is going on beyond that map that has to do with thresholding or region of interest selection or something like that. But then you see this thing, there's these 8 teams here that are correlated with one another, but negatively correlated with the rest. And if we look at their maps, you can kind of see what's going on, right? The maps look inverted, right? For example, here you have activation in the ventromedial prefrontal cortex, which is what we had reported in the original paper for this hypothesis. For this map, for the subjects who are negatively correlated, you see is actually negative activation there. And when we saw this map, what we said was, wow, that looks a lot like just an overall task versus baseline map, right? So you would have this activation, or sorry, deactivation in the ventromedial prefrontal cortex in this kind of default mode area. We looked into what had actually happened and figured out, at least for the one team that hadn't thrown away all of their materials, the other 7, we got their materials and they had all basically mismodeled the task. They did something that is sort of understandable if you don't really know how to model a parametric fMRI study. But it basically made their, it made that analysis be contaminated by overall task activation. Nonetheless, when we take, even including the ones who got it wrong, if we just take everything and put it together into an image-based meta-analysis, you can see reliable findings. And so this has inspired us to start thinking about how can we combine data across analyses to try to, ask which results are robust to choices of analysis. Now there's, people have talked about lots of different ways to try to improve analytic reproducibility. Some are these studies like what we did were called mini analyst studies. Some are using simulation. Some are using blind analysis where you blind yourself or your researchers in the lab to what the actual data mean. So you can't be biased when you're analyzing them. One that we've been particularly interested in the last couple of years is an idea that's come to be called multiverse analysis. The idea here is that you basically come up with a set of reasonable analysis plans, analysis methods, and you apply all of them to the data and you ask how consistent are the results across those different choices, right? So here, this is from the original paper that talked about this by Stegan and colleagues. They show, for some studies you get this is like a histogram of the number of pipelines that give you a particular p-value. So this is saying like most of the pipelines for this, for these two give you p-values that are, down below 0.05. Here you get this wide range suggesting that there really is not a consistent, significant effect there. I'm just going to show you one example of this published in a paper last year by Michael Demidенko in our group that was looking at test-retest reliability. So test-retest reliability is basically if I take two of the same measurements, how correlated are they? Test-retest reliability is essential if you want to look at correlations, like if you want to look at individual differences. One way to think about it is, so the test-retest reliability of a measure puts a ceiling on how well anything else can correlate with it. The idea is like something can't correlate with a variable better than it can correlate with another version of itself, right? Elliott et al. Had a nice paper a few years ago that looked across a bunch of studies in the literature, but also looked across these large data sets and showed that for the most part, test-retest reliability in task fMRI is bad. And so we wanted to know about this, particularly in the context of this task called the monetary incentive delay task. This is one of the tasks that's used in the ABCD study, and it's meant to index kind of reward anticipation. And Michael Demidенko, who joined my lab, had worked a bunch with this task and was very interested in it. And I can talk for a while about why I think it's a horrible task, but I'm not going to do that. He had 3 data sets that we used to try to ask, because if you look across the literature, people analyze these data in many different ways. And we wanted to ask, like, how do those different ways of analyzing the data relate to the test-retest reliability of these estimates of reward anticipation that you get? So we looked across 3 data sets and a bunch of different ways of analyzing the data in the literature, came up with 240 different pipelines, and did this multiverse analysis. So on the top, what you see is like a cumulative plot of just basically the test-retest reliability of the data sorted by that reliability and then showing the variability. So what you see is that at best, the reliability is about 0.4, which is not great, but that's kind of what the literature says where task fMRI reliability should be in general. At worst, it's basically 0. So you would, if you're trying to correlate anything with that, that's basically just futile. There's nothing to be found because the data have no reliability. Then we do this thing called a specification curve analysis, where each row here is one of the things that we varied, that was found to be varied in the literature. These are all choices that people had used in the literature to model reward anticipation in this task. And then they're ordered by their reliability. So the idea is, like, for these where you don't see much difference across the entire, you don't see much clumping at one end to the other, those aren't really strongly related to the measure, like the amount of smoothing or the way that you model motion. It turned out that the thing that mattered the most is what the way that what contrast basically what conditions you're comparing. So if you're comparing this small gain versus neutral condition, you're destined to have really bad reliability. If you're comparing, this large gain versus baseline, then you're like more likely to have higher reliability. It's also a harder contrast to interpret. And similarly, the modeling also makes somewhat of a difference, not as big of a difference. So this shows how differences in, we can use multiverse analysis to help us understand how differences in modeling can make a difference for the results that we get. Okay, I'm going to stop for questions now because I'm about to kind of move on to the next section, which will be focused more on coding. Yeah.
They require researchers to have high intensity for the reliability that helps.
Yeah, let me say, so I'm not sure I exactly, so is the question, would you go use the highest reliability one out of all these 240, or what, maybe you can restate your question again.
And putting them on the higher standard of that's
Yeah, okay, got it. Right. So the question is, like, would having a higher standard for test-retest reliability help with some of the problems that I've been talking about in general? Is that a way to reframe the question? Yeah. So I think that for, like, anytime you're estimating correlations between things, the reliability of those measures is really important. Yeah. And so, knowing the reliability is really important. And then to the degree that there's things one can do to improve reliability, that's really important too. In general, the main thing that we have access to that can improve reliability is just collecting more data per person, right? So if I'm doing a cognitive task and I collect 100 trials, and I get a particular measure of reliability, in general, the more trials, and it's not always true, but in general, the more trials you collect, or the longer in resting state functional connectivity, the longer the imaging runs or the more imaging data you have per person, the higher the reliability is going to be. Yeah, so collecting more data per person is, in general, I don't know if you're going to talk about this, Katarina's going to talk about this, is in general the way to kind of address that. But you'll hear more about that from her. Yeah.
The study with the 20 authors. So when you were asking different groups to identify Russell, so you gave them no criteria, is that correct? Just go with whatever they're doing generally?
Right. Yeah, so we didn't tell them this is exactly how you define ventral medial prefrontal cortex. We just said, go do what you would do in your lab. Yeah.
Okay, and like, even the fact that the whole percentage wise that we were mentioned earlier about, we consider like a 5% change is remarking the fact that, okay, the voxel is important. Not even that was not in the criteria.
So the question is like about what we told people, right? So did we tell them how to define regions of interest? Did we tell them, which is the answer is no. Did we tell them what statistical threshold to use? That's a good question. I don't think we did, but I'm not, we must have said P.05. I know we didn't tell them how to do corrections for multiple comparisons. I'm guessing we told them 0.05, but I would have to look back. I don't remember that.
Stuck in that whole data showing such a crazy result. And so I want to know, since you were part of it, what is the take-home message? Like, do we trust anything anymore? What do we do?
Well, I think the take-home message is that any one study should be viewed as an interesting data point, but not sufficient to sort of thoroughly support or refute any particular hypothesis. And I think this is, in general, like if you look at the medical literature, right, in general, like if you're going to say, should we approve a particular drug, like often that will be based on a single like clinical trial, but in general, what you want to have is like a systematic review across multiple clinical trials, right? Just like we like to have meta-analyses here. Meta-analyses are problematic in lots of ways, particularly because of publication bias. But nonetheless, I think that what it says is that, yeah, you should be very careful about interpreting any particular result, and you really want to see independent replications of the result, or you want to see the data shared so that you or others could go take the data and actually try a bunch of analyses and see how robust the results are to that analytic variability. Yeah.
This is a more general question, but do you have any advice for trainees for how to advocate for more open, reproducible science practices in the lab, especially in a lab that already has established practices?
Yeah, great question. So the question is, how as a trainee can you effectively advocate for using reproducible and open research practices, especially if the lab has like long established practices that are different from that? It's a really hard question. And I know sometimes, trainees end up in a really bad spot. They go to their PI and they're like, hey, look, I went to Seattle for 2 weeks and I learned how to do all this stuff. And the problem is you're going to come back and do it. And suddenly this wonderful paper generating machine is going to grind to a halt because all the questionable research practices that allowed you to find statistically significant results are no longer going to do that, right? And so there are PIs, particularly of a particular vintage, who are less willing to entertain those types of changes. And it's, it can, I certainly know people who have had to leave labs because they just could not convince the PI to change how they do things. And they were so uncomfortable with the way the work was being done in the lab. They knew that it was basically a bullshit generating enterprise. And that, sometimes that's hopefully that doesn't have to be the option for any of you. I think certainly what I have seen in psychology, because this stuff started in psychology 15 years ago, and there were a lot of labs that pushed back hard against the PIs that pushed back hard against pre-registration and things like that. Most of them have come around, in part because their trainees just made it clear to them, look, this is how science is happening now. If you want me to be able to play at the forefront of science, we have to do this stuff, right? And I think that's been helpful. So just making clear, like, look, it's not just me. It's not just me and these goofy people in Seattle. This is how the world is changing. The world has changed. And you know, we need to get get up to speed with how it is that the state of the art says we have to do things now. That's, you know, it's not a great answer, right? It's a really hard question. But I have seen that kind of change work as grad students and postdocs kind of, you know, just push against, you know, on their PI to try to convince them. And that's the argument that I think has worked. Yeah.
I guess most of these hypotheses are like, they look at brain effects. When most of the studies were always an interaction effect, like the visual attention. So usually interactions, like if you have hypothesis interaction about an interaction, you have more like theoretical background to it, but your statistical power is also lower. So like how?
Yeah. So let me restate the question, see if I get it right. The question is basically like, you know, a lot of times we're not interested in a main effect, we're interested in an interaction. And those interactions might have kind of more kind of, you know, theoretical backing. But, you know, how do we deal with that? Is that basically the question? Yeah. So the one thing that we know about interactions is that in general, an interaction is going to have lower power than a main effect. That's kind of generally true, holding, you know, all else constant. So the first thing to say is if you're going to be, you know, if the goal of your study is to test for an interaction, then you need to make sure that your power analysis says that you have a sufficient power to find that interaction, not to find a main effect. Often you see people powering for the wrong test. Otherwise, I don't know that there's really anything special about the kind of effects that you're testing for with regard to the rest of all this. It's really just making sure that your power analysis is aligned with the hypothesis that you want to test.
Are you aware of any meta-analysis that specifically
Sorry, say that again?
Are you aware of any meta-analysis that specifically looked at those hypotheses, like interaction effects?
And right, so the question is, am I aware of any meta-analyses that have looked at specifically at interactions? Not offhand. I don't remember. So like, you know, there's there's several of these mini labs papers where a bunch of different groups collected data on, like, you know, a number of different experiments and look at how they replicate. Some of those might have been interaction effects, but I'm not certain. Yeah. Sorry. Yeah.
The results that the reliability vary across different regions. Because I think to me, it can go different ways. For example, some regions just is known as like for its lower reliability, such as like VNTFC. It can also be understood that maybe these regions just are not involved with these cognitive tasks. So they're just noisier compared to other regions, so they might have lower reliability.
So the question is, how do I make sense of the low reliability of and the variability of reliability in different brain regions in the Elliott paper? And I think you're right that one of the you know, the reliability is going to be related to the signal that's there, right? And if it's a region that either is like, you know, variably activated across people or just really weakly activated, or if it's a region where we know there are signal to noise problems like the ventromedial prefrontal cortex, or you know, where it's harder to get good structural alignment or you know, alignment of the functional data. Those are all reasons that you might expect that. I mean, another thing that could happen, reliability, it makes the assumption that the brain isn't changing, right, across the different measures. There are some brain regions that almost certainly are going to show more plasticity, repetition effects, things like that. So they may show lower test-retest reliability simply because that assumption that the neural function is the same over time is just not valid. Yeah. All right, I'm going to go ahead and move on. Thanks, great questions. If you have additional questions, put them in the Slack, and we'll, you know, try to come to them at the end. Okay, so I was going to do a poll everywhere here, but I didn't get to it. I'm going to do some tomorrow. But let me see a show of hands. How many of you written computer code in the course of your research? Okay, pretty much all of you. How many of you have been trained in software engineering? Okay, small number. How many of you have ever written a test for your code? Oh, wow, actually, that's a good number. Okay. So we think that software errors are a threat to reproducibility. And here's just one example. Jeffrey Chang, a hotshot structural biologist at Salk Institute, published this is all from 2005, 2001, 2005, 3 science papers about this particular ABC transporter protein. He does work to try to understand the structure of proteins. All those papers had to be retracted because they found an error in a piece of an in-house data reduction program, introduced a change in sign. So basically just one sign change totally screwed up the estimated structure. They ended up retracting 5 papers in total. We've also seen this in our own work. So I wrote a blog post about this way back 2013. We were analyzing data from this Open fMRI project, basically trying to look at, could we classify what task a person was doing across like 23 different tasks that they could have been doing. And I wrote this code, Python code. It gave me 93% accuracy. And I was like, wow, if it was 100%, I would have said, no way. 93% seemed too good to be true, but I was like, it's 93. But I gave the code to somebody else to look over to figure out what I was doing wrong, because I was pretty sure something had to be wrong. And Sanmi Koyejo, who then was a grad student, now is a fellow faculty member at Stanford, looked over it and said, oh, look, dude, you did something that is like tiny. Basically, you left out this, right? You messed up how you're indexing those arrays because you left out the colon comma for these 2 things. When you do that, you get 53% accuracy. 53% is actually still great here because chance is down at like, a few percent because it's 1 out of 23. We published the paper with the 53% accuracy measure. But that's a big difference based on 4 characters in the code, right? Here's an even bigger one. We had published a preprint. This went back in like to 2020 or so, 2021, where we were basically looking at the stop signal task data, the behavioral data from the ABCD study. And Patrick from our group is world expert in the stop signal task and identified some issues with how they designed the task that made it kind of problematic to analyze. And this is a statement from the paper for 1,300 out of 7,200 subjects, about 18.3%. This particular type of thing happened that violates this assumption. And the ABCD people, when we put this preprint up and we put all the code up, obviously they weren't particularly happy with us like bashing on their study. So they went and started digging into our code, and Sage, one of the developers for that team, identified an error in this like really horrible looking piece of code right here, right? All these like nested Booleans. Basically, there was like a parenthesis in the wrong place, and it caused it to misidentify or kind of basically to pull out the wrong things. The true number was 447 rather than 1,300, so 6.2% rather than 18.3%. Fortunately, this happened while the paper was under review. We were able to write the journal and say, hey, we identified that we were notified of this problem. We fixed it. Here's an updated version of the paper. The preprint was posted that fixes that problem. The paper still was accepted because this wasn't like a main point of the paper. But it really highlights the fact that, that software errors can have, even in a lab that our lab is, I think, pretty good at this stuff. We make errors and they get into papers. Fortunately, this time it didn't get into the published paper. It just got into the preprint. So here's a big problem, I think. So it's not just that we make errors and sometimes we find them, right? We find them in a way that systematically relates to our hypotheses. So I call this bug hacking, and there's a PhD comics thing that sort of exemplifies this. The idea is that, like, if you have a hypothesis that says this correlation should be there, and you run an analysis and the correlation is not there, you're likely to go make sure you didn't make any errors in your code, right? If you run an analysis and the correlation is like whoppingly significant, you're less likely to go dig in, right? And so, you know, a thing happened in my lab a few years ago where one of the postdocs had a result that was a cool result. We were like working it up. We're going to send in a paper to a fairly fancy journal. But one of the collaborators on it was like, something just seems a little off here. So that collaborator spent about a month digging into the code and the data and ultimately identified an error in the code that invalidated the result. The result that we would have sent into a fancy journal and probably would have gotten published in the fancy journal. And you know, it's great that we didn't publish an irreproducible result. It sucks that these two postdocs ended up without a paper in a fancy journal on their CV, right? So there's this real bind here. Like, you know, you're all incentivized to like get papers in fancy journals, right? Because unfortunately, that's the coin of the realm in academia. But doing all the stuff that I'm telling you about is probably going to make that harder because your papers you're going to be less likely to find inflated results. You're going to be more like self-flagellating, and it's just harder to write really clean papers to send to fancy journals when you've actually done the right things. I think it's changing. I think it's becoming easier I think those journals are becoming more open to like letting everything show, but nonetheless, it's, you know, I just have to be honest about the fact that, like, you know, the stuff that I'm talking about is in conflict with a lot of the incentives that you have. And that's a hard problem that, you know, many of us are working on at kind of high levels, and I'm happy to talk more about that. But it's just, it remains a bind. So continuing with ways to make your own life even more difficult, is this what I call the principle of assumed error. Whenever you get a good result, assume that you did something wrong and go try to figure out what you did wrong to get the good result. Try to figure out how you can validate it. Sometimes that might be getting somebody in the lab to write completely independent code, or it might be going and getting 3 different AI systems to write code and to do the same analysis and seeing, or at this point, it might be giving everything you've done to an AI system and saying, tell me what I did wrong that could give me this result. Right now, AI, I think, has really opened up, I mean, it's opened up a whole mess of things that are really a mess, but it's also opened up the ability for us to critique ourselves in ways that I think are really useful. Okay, so what do we want to do if you're learning how to write code? And I'll talk tomorrow about, you know, how this relates to using AI to write code. We want our code to be good. What does good mean? Good means readable, right? The most important thing about good code is that it's readable, because, you know, most people, you know, the code gets read a lot more than it gets written. It needs to be robust, meaning that it like it does the right thing when it should do the right thing, and it doesn't do the wrong thing when it shouldn't do the wrong thing. It needs to be modular, which means that basically it's broken into a lot of pieces, and there's a number of reasons why modular code is good. I'm happy to talk about that. And it needs to be well tested. And you know, it's funny, I hadn't updated this slide. I think I would say this a little bit differently. I'm going to talk more tomorrow about coding in the age of AI. And coding is a craft, right? Coding is a skill. And the only way to improve it is through consistent and deliberative practice. I think increasingly people aren't getting practice at writing code from scratch. They're more getting practice at getting a model, a language model to write the code for them, and then kind of seeing if it works. It remains to be seen whether that's a good way to learn how to code. I mean, it is how people are learning to code now. Because I think that, you know, as you'll see tomorrow, the problem is that you know, the better the tools get, the harder the problems are to solve where they fail, right? And so you need to know, you know, really know how to read code and understand code in order to be able to figure out, why isn't this model doing this thing correctly? And so I think, you know, I think, you know, we're doing a huge experiment on people right now as a society, right? Giving them these models and saying, hey, you know, go learn programming. And of course, people are going to use the tools to write code. But I'll talk tomorrow about how I think that, like, the writing of the code is not necessarily the most important part of learning how to program. So we'll come back to that. I think coding style is really important. And this is mostly because being able to read code is really important. You want to be able to look at the code and know what it's doing. And fortunately, the AI systems actually generate pretty good, pretty readable code, and can, if you help them, they can do an even better job of it. These are all really good books. If I had to pick up one, I'd probably say The Art of Readable Code. It's pretty short, pretty succinct, and basically goes through all the things about like what makes code good. The other is John Osterholt's Philosophy of Software Design, which really goes into details about why do you want your code to be modular? Why does it matter? Things like that. Like what, how is it, designing the way software works? What are the principles? So often you'll see people write something on social media about like, oh, I'm about to submit my paper, but I need to go back and comment my code because it doesn't have any comments in it. And there's often this idea that like what makes code good or not is whether it has a lot of comments in it. And if you read the software engineering literature, it's basically exactly the opposite of that, right? That really good code is so clearly written that you don't need any comments to know exactly what it's doing and exactly why it's doing it. And the comments should be there to help the reader understand the things that they can't get from reading the code itself. So just a couple of quotes. Clear and expressive code with a few comments is far superior to cluttered and complex code with lots of comments. Or from this famous Kurting and Poldrack book, don't comment bad code, rewrite it. And now it's basically, don't comment bad code, have Claude or GPT-4 rewrite it for you. There's a lot of tools for like looking at the quality of code. So there's particularly tools like what we call static analysis tools. These can basically like find errors, change, fix style. What's the name of I'm trying to remember the name of the newest one that I'm using now, but I can't. I'm blanking on the name of it. Anyway, there's a really good new one. But anyway, these tools are great to use just to make, like, even if your code is like messy, you can give it to these tools and they will basically make your code prettier. Language models can do that as well. So code review is another thing that, as I'll talk about tomorrow, has gotten a lot easier with language models. It used to be the case you had to go find an expert Python programmer in the lab to like look at your Python. And we know that that having somebody else review your code is really important. We do it in my lab occasionally where we'll have a kind of a public code review. Somebody will bring their code and I or somebody else will kind of walk through it. And it's mostly just asking them, like, what are you trying to do here? Why are you doing that? Or what does that, obscure variable name mean? Why do you have a number in there and I don't know what that number is, things like that. And it's been a great way for our lab to kind of just, for people to learn kind of good, clean coding skills. But as I said, I'll talk about tomorrow about how AI has become really good at this as well. Okay, so we think that having tests is really important, and especially with AI generated code. But there's different types of tests. The most general one is what we call a smoke test, which is basically like, does the code run without crashing? Like, if you plug it in, does it start smoking? A sanity check, like are the values within reasonable ranges? And then usually when people talk about testing, that what they mean are unit tests, which are basically like tests of particular parts of the code to make sure that they're doing the right thing. Here's an example of somewhere where some testing could have really helped someone. This was a paper published by this group about conspiracy theories and rejection of science. And they had originally claimed that there was not a significant correlation between age and these other variables. But the data set had two notable age outliers. This is supposed to be just adults. Ages 5 and 32757. And so the results changed. So they had to issue a correction to the paper. They could have very easily added an assertion in their code that said, let's just make sure that the ages are, assuming that the ages were supposed to be between, say, 12 and 120, let's just make sure that the ages are in the range that we expect them to be in. And that would have saved them because it would have said, hey, there's something wrong here. So whenever there's something that should be true, like, reaction times should never be negative, right? Ages should never be 32,757 and so on. Put tests in the code for that to make sure that if something weird is going on, you catch it. So we've we're trying to move to an approach that's come to be called test-driven development. And for my personal projects, I've started using it and find it really useful. And especially when I'm working with AI tools, it's really useful. The idea is that before you write any code to solve a problem, you write a set of tests that will basically define when the problem is solved, right? The problem is solved when the tests pass. So you initially write a set of tests that fail. And then you write the code that's required to make the test pass, and then you stop. And that helps prevent, there's this thing that in the software engineering literature people call gold plating, which is basically writing more code than you need to write to solve the problem. And so test-driven development both helps you make sure that you're actually solving the problem, but also prevents you from kind of over-solving it. And then obviously you can kind of go back and do more. I'll talk more about this tomorrow. And then now that there are great tools for automated testing. Once you've created your tests, now whenever you push your code up to GitHub, you can have it automatically run the tests. There's a thing called GitHub Actions that makes it pretty easy to do this depending on your code and your data. So you can choose a workflow. It'll help you put together a test thing, and now it will go and in this case my test failed because I don't know why, but it's a big red X. And that's really useful because then you can basically make sure that if those tests get run every time you check in new code, then if you do something, even if you aren't running the tests all the time, if you do something that breaks something that you did last month, the test will fail and you'll know that. We call that a regression. So I'll mention this now and I'll mention it tomorrow. I'm writing a book, like kind of an open online living textbook right now called Better Code, Better Science, which is all about basically doing better scientific software engineering, kind of realizing that most scientists today are amateur software engineers, and we know that that's a recipe for errors. So it's going, and it's a growing textbook. It hasn't really gotten to the meat of the AI coding stuff so far. And I have been releasing like a section every week, but this week in honor of NeuroHackademy, I'm releasing a section every morning. So via my Substack. So the book all the stuff is online, the book, but if you want it in like daily or weekly bites, just go sign up for my Substack and you'll get an email, every day this week. And then in the weeks coming with like little sections from the book. This week's stuff is all about software testing. I started software testing in the last couple of weeks, and then there's a bunch more this week. So check that out. I just want to make one final point. Talked a lot about reproducibility, right? The idea that if we do the same thing more than once, we should get the same answer. But we know that, what we really care about is not just that we get the same answer, but that we get the right answer, right? And so in psychology, we talk about reliability versus validity, right? Reliability is the idea that you get the same answer each time. Validity is that you get the right answer, right? So in this case, you could be getting highly reliable answers, but you're not hitting the bullseye. And so we need to care about whether our code's getting the right answer as well as giving us, the same answer. That's often harder to do. And often requires learning how to run simulations. Now, again, AI is going to be pretty good at helping you generate simulations, but you're going to need to know what you're doing in order to really make sure that they're doing the right thing. So one thing that we like to do is what we call parameter recovery. Like if I've generated code that should be able to find an answer, I want to give it data where I know what the answer is and make sure that the answer it gives me back is the one that I put in. Like if I inject a signal, it should give me back that signal. If I just give it noise, it should, I run it 100 times, it should give me 5% yeses if it's a 5% false positive rate. And that's what I mean by null randomization testing. And there's a lot of tools for generating simulated data out there. Synthetic data vault's one of them, but there's a lot of others. Okay, so just in conclusion, I hope I've convinced you that we have a problem and that it's worth trying to fix it. It's not specific to neuroscience or psychology or cognitive neuroscience. We're seeing the same problems in many other domains of science, especially where there are like really complex data and complex analytic workflows. I think the statistical power is like the thing that is probably most important for solving this problem, because so many issues just go away once you start finding results once you start finding estimates that are actually stable. But it's also the case, I think, that doing a better job with software engineering is going to help us improve the way that science works, and you're going to learn a lot of good stuff this week to know how to do that. And finally, I just want to thank all of my friends and family and people who gave us money to do this stuff, and got a few minutes for questions.
Questions?
So yeah, questions in the room, and I don't know if anybody can look on Slack and see if there are any there.
Kind of a general question, but like, if you're looking to create like a new hypothesis or like a pilot experiment, and you're reading all these literature that are unreliable, like what kind of skepticism or like how would you think this data is reliable?
Yeah, so the question is like, what do we do? Like, you're a scientist, you want to like go run some studies, and you have to base those on a literature that may or may not be reliable. What do you do? I mean, the first thing I will say is that you always want to make sure that you can replicate the finding that you're building on. So I've had two people in the lab who basically started down a road of trying to build on a published finding, and ultimately realizing that the basic effect that they were trying to build on was not reproducible, even though it had been presented in multiple papers. We actually just have a registered report coming out that started, I think, 6-7 years ago. It basically started pre-COVID, and then COVID slowed it down. This registered replication thing across multiple labs for one of these, where he was then a postdoc, Patrick Bissett is now a research scientist in the lab, wrote a fellowship to go build on this published finding, and then started trying to build on it and realized that he couldn't get the basic result. And now we know that we've done this registered replication. We have a bunch of studies in the lab. We know that the result is just not there. Another person in the lab had similar experience, ended up leaving science, I think in part because he was so demoralized by the fact that that the thing he was building on wasn't reproducible. And actually what was really frustrating is I ended up talking to another big person in that field, telling them about how we failed to replicate that finding. And they said, oh yeah, we failed to replicate that a while ago. And it's like, why didn't you write that down and tell people, So it also is, I think it's really important to publish, publishing null results is hard. And it can also really piss people off when you're saying that what they found is not a reliable effect. But it's really important to prevent trainees from building a whole experimental edifice on something that's just total, crumbly sand. Yeah.
Oh, I have this specific, but I'm wondering how these principles translate to research with non-human primates. So for example, you mentioned minimum 20. So that's often really not.
Yeah, great question. So the question is, how does this translate into domains where in general you have very small sample sizes, like non-human primate work being an example? I'm guessing, again, you're going to say something about this. When is your talk?
Yeah, 3:30.
Okay, 3:30 today. So Katarina's going to say more about this, but I'll give you my view, which is that there are different ways, that there's not one way that science has to happen, right? Now, if you care about finding individual differences between people, then you have no choice but to go get large sample sizes because you need to see the entire distribution, right? But that's not the only types of questions you can ask. So the strategy that people in the non-human primate world use, right, is to collect a lot of data per animal. They'll maybe do it on 3 or 4 animals and treat those as independent replications, and also maybe do replications within the animal as well. That's a perfectly reasonable thing to do, as long as you write it in a way that doesn't make claims about generalizability that aren't supported by the statistics, right? If you want to say we can generalize out to a population, but you haven't sampled that population and you haven't quantified, how the variability with respect to the population, then you can't make those kind of claims. But you can certainly make claims, as people in that field have done, that turn out to be robust and reliable. So I think there's just there's different ways of doing science, and it's similarly if you look at like the rodent world, like people who do, you know, say like, you know, electrophysiology, optogenetics, all that sort of stuff in rodents, often you'll see that like within a paper, another strategy that they use is like, you know, you might have like, you know, the main experiment, then like 8 or 9 control experiments, right? All of which are like tweaking one little thing and making sure that it behaves as it should. That's another way to do it. That's not a way that we generally do it in human work, in part in human neuroscience, because it's so expensive to collect, you know, individuals. But yeah, one just has to make sure that the nature of the results, that they're robust in whatever way they're supposed to be robust, and that your claims are calibrated with respect to the design.
That's the like level of resolution. So if you're using visions in like a, you know, 3T or 7T or 9T or like ultra high field MRI, how does the resolution change the nature of the types of issues that you're considering?
Right. So the question is like, how does the nature of the measurement, for example, the field strength at which you're doing MR, how does that change the kind of the analyses or the claims or the you know, how this relates to reliability? And I don't think there's a single like one size fits all answer, right? So for example, moving to higher field strength in general is going to give you more signal noise, at least for some types of questions. It also is generally going to give you like worse artifacts. And, you know, like one thing I think that's happening now in the high field MR world is that because the tools for high field MR are not as well established, it's a little more kind of Wild West, like a lot of groups just do their own thing. And it makes it harder to know like how reproducible that stuff is. That's emerging, obviously, but so I think there's trade-offs. Do we have time for one more? Or? Okay, one more. Yeah.
In the replication and prediction study, if I heard you correctly, you said that among the groups that didn't make that mistake, the brain imaging maps had quite high correlations to each other, but they came to different conclusions on the hypotheses. You mentioned like thresholding, difference in thresholding could be one reason. I'm just curious what the other reasons were, and if they were more than technical in nature, or were they more just to do with how the researchers like interpreted similar values? Maybe?
I don't. They shouldn't be up to that level of interpretation. There should be a technical explanation. Unfortunately, it was very hard to find that because, you know, if half the groups had done it one way and half had done it another, then we could do a direct comparison. But because everybody did things so differently, there were a few things that we could like quantify and ask, is this related to, you know, is the amount of smoothing related to, you know, the finding or something like that. But in general, we couldn't do that because there were just, the analytic landscape was so big. Yeah. So unfortunately, I don't know the answer to that. Cool. All right. Well, thanks.